President of Microbiologists Society India and a former professor and head of the Department of Microbiology, Baba Saheb Marathwada University, Maharashtra. Most welcome, sir. Yes, Amitabh sir, एक बार माइक को म्यूट करिएगा प्लीज.
बिल्कुल.
जी जी जी. Yes, Deshmukh sir, your speech. Over to you. Your speech.
Thank you.
Yes.
Thank you. Today I am very happy to play a role of a special guest. The reason is that the organizer is a human rights foundation along with the Jan Jagran Sansthan. The topic is a stem cell technology, which is really a burning topic. And probably if you ask me which topic will dominate the world in the future, it is the stem cell technology. And the speaker, whom I know personally, she was working in Kolhapur. From that, she was regularly in contact with me. She is very enthusiastic, and I know that among the young generation, I am having good hope from Indumathi. So today, it is my duty to wish all the best for today's her speech. As this lecture is being recorded, it is also my duty to circulate among 20,000 students. So definitely, slowly by slowly, people and students will take advantage of the today's topic, today's discussion. And along with me, one of my friend, Dr. Swarnajit Singh sir, he is here. I also welcome Dr. Swarnajit Singh for this event. And Dr. A.M. Prasad, who is a pillar behind today's organization, today's event. I appreciate his efforts and will say that, sir, whatsoever you will do in science, Microbiology Society always will support you. And Sarat Surwa, one of my best students, he is also doing well in Jharkhand. So my blessings are always with him. I wish all the best for today's program. Thank you very much.
Thank you so much, sir. Thanks a lot for your encouraging words, and your blessings are always important for us to proceed for this program, for any program, for anything, in fact. And thanks a lot, sir. Yes, I would request our guest of honor, Dr. Swaranjit Singh, sir, to address the audience. Over to you, sir. Most welcome, sir. Most welcome.
Thank you very much. I am very happy today to be here in the stem cell session. Stem cell technology is a burning topic nowadays, as Dr. Deshmukh said. Stem cells have the potential to become any type of cell in the body. We have 100 trillion cells, and those are in different types and shapes. Lifetime of the cell is generally 90 to 120 days, depending on our age and metabolism. There are over 200 cell types in the human body. Each type of cell is specialized to carry out a particular function, either alone, solely, or by forming a tissue. Stem cells protect us from UV light radiation. They boost immune system, control diabetes, help in weight loss, help in digestion, and in better circulation in our body. Today, I'm very happy that Dr. Indumathi S is giving a talk on stem cells. I have interacted with Dr. Indumathi in a recent talk seminar on MBSI site. I find her very brilliant, intelligent, and well informed. So today we will be very benefited, especially the students all over India and abroad, by the talk of Dr. Indumathi. Indumathi, all the best.
Thank you, sir. Thank you for your words, for your blessings for this program. And yeah, so today we are having this lecture on stem cell technology, and we have Dr. Indumathi Somasundaram Madam from Chennai, Tamil Nadu, India. She is our chief speaker, and we can consider her as the keynote speaker for this topic. Actually, she is the stem cell researcher in herself, so she'll be delivering her lecture. But, ma'am, I request you that there is one student speaker also. So she is having a short presentation, very short presentation. So she'll be delivering the presentation, and after
Sure, sir.
listening to her,
Yes,
we
yes, sir.
will
It's a pleasure
proceed.
listening to students. Please go ahead.
Yeah, ma'am. Okay. So, Shruti,
Sharat ji, we should welcome our chief guest also, Dr. A.M. Prasad ji.
yes.
A.M. Prasad ji,
Ma'am,
हाँ,
yes, sir.
yeah, we
Yes,
should.
sir. Sir, please,
I have thank you very
Maheshwari,
much,
sir.
Amitabh ji. Thank you very much. I
Maheshwari, sir,
am
please, sir. Yes, sir.
my ears are glued, and I feel extremely fortunate that in recent times I have missed some, but this is such an important topic, and we are fortunate in having one doctor. I mean, having Dr. Indumathi ji, about whom words of praise have already been showered on her. And after hearing those words of praise about her, I'm not able to check my own sentiments and add to what has been said about her, that there are very few people in the world who are working with dedication for the welfare of people. I'm sure that this technology, I have heard this technology is the name of this technology for some time. I'm sure this must have arrived at new landmarks in its journey of research, and it must be nearing the expected outcome of the efforts of researchers like Dr. Indumathi ji. With these words, I welcome her again and thank Amitabh ji for having made this forum so invaluable. And it is your infinite, if I may say, and unparalleled commitment which has made it possible for us to keep learning every Saturday and Sunday. God bless you, and God bless Dr. Swadeep ji, Dr. Indumathi ji. Please go ahead the formality is over. I am grateful for keeping me
वहाँ पर
in the
लगने से
loop.
खराब
Very
ही नहीं
kind of
लगा
you. Thank you so much. Bye.
Thank you, sir. I'm very happy today to get the blessings of such a great scholar, Dr. M. Prasad, also. So we are lucky with your blessings and with Almighty's blessings. I'm conducting such courses, such webinars every Saturday and Sunday. And we are booked up till 12. And so many people are requesting again and again that they want to speak. And there was a ring from abroad also. So we will be arranging with your blessings. Thank you, sir.
Definitely,
I
sir.
seek permission from Indu ji to have a Guru Bandana. इंदू जी
Okay.
मैं गुरु वंदना कर दूं, आधा मिनट
बिल्कुल
लगेगा
बिल्कुल हाँ बिल्कुल 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 कर दीजिए
प्लीज. ओम गुरुर्ब्रह्मा गुरुर्विष्णु गुरुर्देव महेश्वराय गुरुर साक्षात परम ब्रह्म तस्मै श्री गुरुवे नमः. अखंड मंडलाकारं व्याप्तं येन चराचरं तत्पदं दर्शितं येन तस्मै श्री गुरुवे नमः. अज्ञानतिमिरान्धस्य ज्ञानाञ्जनशलाकया चक्षुरुन्मीलितं येन तस्मै श्री गुरुवे नमः. थैंक यू.
थैंक यू सर थैंक यू नाइस प्रेयर नागेश जी आपने वातावरण शुद्ध कर दिया ओके नाउ शरद जी
जी जी जी जी जी बिल्कुल सर. धन्यवाद सर. धन्यवाद नागेश सर. धन्यवाद महेश्वरी सर. शुरू करते हैं. श्रुति स्टूडेंट स्पीकर है. ये बायोटेक्नोलॉजी डिपार्टमेंट की अंडर ग्रेजुएशन की स्टूडेंट है. तो हमारे बीच ये स्टूडेंट स्पीकर है. हमें मौका देना चाहिए स्टूडेंट्स को भी और उनको सीखने का भी मौका मिलता है इससे. बड़े बड़े एक्सपर्टीज से जैसे हमारी डॉक्टर इंदुमती मैम है. इटसेल्फ आज का जो टॉपिक है वो उनका ही फील्ड है. She is the expertise in stem cell technology. So we have invited Dr. Indumathi ma'am. Most welcome, ma'am, and we are grateful to you, ma'am. So yeah, Shruti, please start presenting your presentation, and yes, ma'am will give remarks to your presentation, of course.
All
Yeah,
the best,
Shruti.
Shruti.
Yeah,
All the very best.
yeah. Quickly present your presentation and start with your lecture.
इसमें क्या करोगे बताओ. ये हमारे इसमें भी रहेगा. अब इसमें हम एक कोई भी हमारा निकाल लो.
Okay, myself Shruti Savule. I am from semester Kasturba Walchand College, Sangli. I am taking an bachelor's degree in biotechnology. Today I will give an webinar on stem cell cryopreservation. Then, first, what is stem cell? Stem cells are undifferentiated cells with the potential to develop into various specialized cells types in the body. Stem cells have ability to develop into new cells and entire organs. स्टेम सेल्स यह ऐसे सेल है हमारे बॉडी में जो हम न्यू सेल्स टिश्यू और ऑर्गन्स प्रोड्यूस कर सकते हैं. देन क्रायोप्रिजर्वेशन एक बायोलॉजिकल टेक्निक है जिसमें सेल्स टिश्यूज एज वेल एज होल ऑर्गन्स प्रिजर्व किए जाते हैं अंडर लो टेम्परेचर इन लिक्विड नाइट्रोजन. Then, here are the steps for stem cell cryopreservation. First is enrollment. After choosing the right stem cell bank, signing up for cord blood banking service will before the accepted date of delivery is done. Following are the enrollment processes. Will receive a collection kit which needs to be carried to a hospital at the time of delivery. This kit contains the tools and instruments required for the collection or umbilical cord blood tissue and maternal sample. So first स्टेप इनरोलमेंट में जो वुमन है वह राइट स्टेम सेल बैंक चूज करने के बाद उसके साथ सब पेपर वर्क डन करने के बाद स्टेम सेल बैंक उसको एक कलेक्शन किट प्रोवाइड करके एज आई शो इन पिक्चर। वह कलेक्शन किट उसको उसे डिलीवरी डेट के पहले मिल जाती है। उसमें जो भी कलेक्ट स्टेम सेल्स कलेक्ट करने के लिए इंस्ट्रूमेंट्स लगते हैं वह उसमें होते हैं। Second step is a collection. After the baby is born, the umbilical cord is clamped and both the ends and cut by the attending gynecologist through a safe procedure. The sample collected from the umbilical cords are a little amount of mother's peripheral blood inside the umbilical cord, small pieces of umbilical cord tissue. These samples are collected by the paramedic and stored in the collection kit, which are sealed properly and sent to a lab for processing. तो ये सेकेंड स्टेप है कलेक्शन स्टेप में आफ्टर द बर्थ ऑफ बेबी जो मैटरनल ब्लड और बेबी का यूम्बिलिकल कॉर्ड है वह कलेक्शन किट में कलेक्ट किया जाता है। उसमें मदर्स पेरिफेरल ब्लड एज वेल एज स्मॉल पीसेस ऑफ umbilical cord of tissue बेबी का कलेक्ट किया जाता है। सेकेंड स्टेप है ट्रांसपोर्टेशन। The collected samples must reach the laboratory within 48 hours and usually transported through a special gynecologist provider to ensure faster delivery and avoid X-ray screening. Some stem cell banks offer online tracking facility to the customers in order to keep track of the sample transport. तो कलेक्शन किट में सैंपल कलेक्ट करने के बाद वह कलेक्शन किट स्टेम सेल बैंक की ओर ट्रांसपोर्ट की जाती है। वह एक अंडर स्पेशल गायनेकोलॉजिस्ट प्रोवाइडर के थ्रू ट्रांसपोर्ट की जाती है फॉर अ सेफ्टी। और सम स्टेम सेल बैंक हमें ट्रैकिंग फैसिलिटीज भी अवेलेबल करके देती है। उसके बाद नेक्स्ट स्टेप है टेस्टिंग एंड प्रोसेसिंग। Once the samples reach the laboratory, they are tested for contamination or infectious diseases. The tests are also performed to check the cell count, viability, and sterility of the sample. The cord blood sample is processed by automated processing technique to remove the red blood cells and retrieve only stem cells. The cord tissue samples is cut into small pieces and the stem cells are isolated after processing. They undergo various levels of quality control assessment to ensure the viability and the purity of stem cell. तो स्टेम सेल बैंक में स्टेम सेल्स आने के बाद सबसे पहले उसके फिजिकल एग्जामिनेशन होती है। फिजिकल एग्जामिनेशन के वक्त उसमें कुछ कंटेमिनेशन या इन्फेक्शन या डिसीज है क्या वो डिटेक्ट किए जाते हैं। उसके बाद उसका सीबीसी वायबिलिटी और स्टेरिलिटी टेस्ट होता है। उसके बाद स्टेम सेल यूम्बिलिकल कॉर्ड में वेरियस टेस्ट किए जाते हैं और उसमें से स्टेम सेल्स डिराइव करके नेक्स्ट प्रोसीजर की जाती है। लास्ट है प्रिजर्वेशन। तो umbilical cord as well as maternal blood और peripheral blood में ऑल टेस्ट करने के बाद वह प्रिजर्व किए जाते हैं अंडर क्रायोजेनिक टैंकर्स इन लिक्विड नाइट्रोजन इन फ्रोजन कंडीशन। Here are the picture which I explained. Then methods of stem cell cryopreservation. तो यह जो पिक्चर है यह लैब है स्टेम प्लस क्रायोप्रिजर्वेशन सांगली जहाँ पे मेरे एक्चुअली इंटर्नशिप कंप्लीट हुई है। मैंने उसमें से ही लैब के पिक्चर्स इसमें ऐड किए हैं मेथड्स में। तो फर्स्ट है फिजिकल एग्जामिनेशन लैब। इस लैब में जो सैंपल आता है उसका सबसे पहले टेम्परेचर, वेट और ब्लड कोगुलेशन चेक किया जाता है। अगर उस सैंपल में ब्लड में कुछ कोगुलेंट्स रहते हैं तो उसे रिमूव किया जाता है और ओनली लिक्विड ब्लड लिया जाता है फॉर प्रोसेसिंग। नेक्स्ट लैब है ट्रांसफ्यूजन ट्रांसमिसिबल डिसीज स्क्रीनिंग लैबोरेटरी। तो इस लैब में जो सैंपल आता है उस मैटरनल ब्लड में से सीरम सैंपल सेपरेट किया जाता है और उसमें कुछ डिसीज एंटीजेंस और एंटीबॉडीज है क्या वो डिटेक्ट किए जाते हैं। उसके बाद थर्ड लैब है हिमेटोलॉजी एंड सीरोलॉजी लैबोरेटरी। तो इस लैब में ब्लड का टोटल सीबीसी काउंट किया जाता है कि ब्लड में कितने प्लेटलेट्स, आरबीसीज और डब्ल्यूबीज प्रेजेंट है वो चेक किए जाते हैं। उसके बाद उसमें उसकी वायबिलिटी भी चेक किए जाते हैं कि उसमें कितने सेल्स लाइव है, कितने डेट। उसके बाद फोर सॉरी उसके बाद फोर्थ लैब है स्टेरिलिटी टेस्टिंग एंड मीडिया प्रिपरेशन लैब। तो इस लैब में जो ब्लड सैंपल आता है वह फंगल और बैक्टीरियल सैंपल के साथ टेस्ट किया जाता है फॉर अ रिसर्च पर। देन फिफ्थ लैब है कॉर्ड ब्लड प्रोसेसिंग लैबोरेटरी। इस लैब में जो हमारा उम्बिलिकल कॉर्ड जो है वह सेपरेट किया जाता है और उसमें से स्टेम सेल्स टिश्यूज सेपरेट करके स्टेम सेल्स डिराइव करके उसमें प्रोसेसिंग करके उसे स्टोरेज करने के लिए नेक्स्ट लैब की तरफ भेजा जाता है। और लास्ट लैब है क्रायो स्टोरेज रूम। क्रायो आफ्टर स्टेम सेल्स डिराइव होने के बाद ऑल प्रोसीजर होने के बाद स्टेम सेल्स इस लैब में प्रिजर्व किए जाते हैं अंडर क्रायो अंडर लिक्विड नाइट्रोजन इन लो टेम्परेचर। क्रायोजेनिक टैंकर्स में वह स्टेम सेल्स प्रिजर्व किए जाते हैं। तो स्टेम सेल्स के एडवांटेजेस, umbilical cord blood has a potential to cure over more than 100 diseases। यूम्बिलिकल कॉर्ड ब्लड जो होता है उससे हम 80 प्लस डिसीजेस क्योर कर सकते हैं। मोस्टली जो ब्लड रिलेटेड डिसीज है वो 100% क्योर किए जाते हैं विद द हेल्प ऑफ स्टेम सेल्स। सेकेंड है cord blood is readily available when needed if collected and stored। जो कॉर्ड ब्लड है वो हम कलेक्ट करके स्टोर करें तो हाँ वो इजीली अवेलेबल हो जाता है जब उसकी जरूरत होती है तब। थर्ड एडवांटेज है cord blood stem cells are youngest and most adaptable form of cells and the body more readily accept the cells into the system। जो कॉर्ड ब्लड में स्टेम सेल्स रहते हैं वो सबसे यंगेस्ट होते हैं और हमारी बॉडी वह इजीली एक्सेप्ट करती है। ये उसका एडवांटेज है। लास्ट एडवांटेज है स्टेम सेल हेल्प्स रिसर्चर्स फॉर बेटर अंडरस्टैंडिंग ऑफ डिसीजेस। तो विद द हेल्प ऑफ स्टेम सेल्स रिसर्चर्स के लिए स्टेम सेल्स एक हेल्प करता है फॉर अंडरस्टैंडिंग ऑफ वेरियस डिसीजेस। ओके, थैंक यू।
Very nice, Shruti. Nice presentation. Uh,
थैंक यू भैया।
yeah, I hope ma'am should give remarks to your presentation, and of course, after that, we are eager to hear Dr. Indumathi ma'am. Yeah, ma'am, please over to you. Now the stage is all yours for 1
Yes,
webinar
yes,
complete.
yeah. So, Shruti, before I speak about your presentation, thank you so much to all the dignitaries about Jan Jagran Foundation and Human Rights Association. A special thanks to the entire team, Amitabh sir, Swaranjit sir, Deshmukh sir, who has always been a great pillar to all of us, and you know, I'm also grateful to Sarat Sufra sir, right? I hope I pronounced your name right. So I'm so excited
Ekdam
and
correct.
thank you, sir. So Shruti, great. So what are you into? Which year are you are in?
Now I am completed second year in biotechnology.
Excellent, excellent. So I'm so happy because we were introduced to stem cells when I was in my master's second year end that we had a paper. That is where I got interest on stem cells. And now it's so good that, even in UG, we are talking about cryopreservation, and your presentation was crystal clear. It was so good. You were confident. There's no flaw. And you presented excellent. Your knowledge at this particular thing,
Thank
your
you so
preparation. So
much.
this shows that you have taken effort to really prepare, understand every step of cryopreservation, starting from, how we have to collect the sample to freezing to thawing to the long-term cryopreservation to everything. That was really amazing. And I hope that you take it so much forward and don't stop this interest. Let the curiosity get going on.
Yes, yes, ma'am.
Keep
I actually
participating. Keep
completed my
participating.
internship.
Yes, Shruti.
Yes, ma'am. Yeah, actually, I'm completed my internship in STEM plus bank, so I'm get interested in STEM stuff.
Good, good. All the best. So nice.
Thank you.
Yes, ma'am. Yeah, most welcome,
Can I?
Indumathi ma'am. Please
Yeah, yeah.
proceed.
Okay,
Please
okay.
proceed.
I'll just share my screen. So, very good evening to all the participants. My utmost gratefulness to our chief guest. Uh, you know, I hope I'm pronouncing well, sir. Akauri Maheshwari Prasad, sir. AM Prasad, sir. I'm seeing you for the first time here, sir. So, I'm so grateful that you are here. I'm grateful that AM Deshmukh, sir, Swaranjit, sir, all are listening to my talk. So, namaskar, sir. Thank you for joining us.
Namaskar.
Thank you for joining us, sir. And so much thanks to all our participants who are eagerly looking forward to. Yeah. So, is my screen visible?
Yes,
Is my screen visible? Okay. Are the slides changing?
not yet.
Okay, just a second. Why my slides are not changing?
मैम वो स्लाइड शो पे आप आ जाओ, स्लाइड शो पे एनिमेशन के बगल में जो ऊपर में जो स्लाइड शो दिया हुआ है.
No, no, I am into slideshow. Now is it? Are you able to see the slide changing?
इसमें लाइब्रेरी
Not yet,
डिवाइस
ma'am.
लिखना था.
Are the slides moving?
Slide is
Not
not
yet, ma'am.
moving. Not moving.
I I have shared the window, I've presented the screen. I'm able to do it here, but I'm not able to do any changes within the screen. Not sure. Just give me a moment. I've shared the
श्योर श्योर
window. I'm sorry, sir. Just give me a moment.
श्योर मैम.
I'll try and I think better I'll go to entire screen and then from there I can. Is my presentation seen now?
Presentation is visible. Yeah, it is visible. Yeah,
Moving.
Is it
slides
now
are also
changing?
changing. Changing,
Okay.
changing,
Okay,
Yes,
perfect.
changing.
ma'am.
So
It's
I
changing.
think you're able to see the entire slides because I've shared the entire screen, but I think the entire
Very
slide.
clearly, very clearly,
Okay,
very
great.
clearly.
Great. Thank you, sir. Thank you for this. Sorry for this short glitch. So the topic why I said series of stem cell biology because stem cells are something very booming and it's need of the hour. And why I said series is because of great news which I wanted to tell and start with. The NAC, ICSCRT, and the Stem Cell Ethical Committee, which is ICSCRT team, there's one team who always sits behind and gives some ethical approvals. There's a lot of ban in India. So recently, that particular team, the central government, they have dissolved this team in a notion that like other foreign countries, India should grow more in the field of stem cells, and that is the reason this entire ethical committee team, which is NAC, SCRT, and ICCRT, is being dissolved, and that is a great news for this year. And now individually we know that we follow the integrity ethics and the protocol, and based on that I'm sure that every one of us will follow those ethics and integrity and take stem cell to the next level. That's why this is just not the start, but it's a series. We want every one of us to get into this field. And now what you're seeing in this screen, if the slides are not changing, please let me know. Is it changing? Are the slides changing?
मैम
Yes, ma'am.
अभी
Yes, ma'am.
yes,
Yes,
yes.
yes,
Okay.
it's okay. Okay, good.
Yeah, thank you. So if you see this first slide, I've put a lot of images. That's because everywhere you have stem cells. So I just wanted to, create the curiosity of the students to say that the stem cell starts from zygote. So basically the potential of stem cell is something where a life begins. So when the male and female gamete fuse and the zygote starts, and then it becomes morula, then it becomes blastocyst stage of the embryo, and from there the stem cell starts. So as you see here on the slide, this is the blastocyst stage of the embryo where the inner cell mass, after getting implanted into the uterus, becomes the embryonic stem cell, and the outer covering is the placenta, where you can see this is how The embryo grows, this inner cell mass becomes the embryo, and the outer covering is the placenta. And this is a pluripotent stem cells, because we're going to see about differences between different stem cells. I've kept it very minimal, basic, and a complete coverage of everything. So बहुत सारे students को इस field में interest आए and they get into this field. So pluripotent stem cell is, pluri means many, proti means whole. So the complete zygote, the inner cell mass stage, which is there, that is a pluripotent stem cell, because if you see that embryo is giving rise to all three germ layers, ectoderm, mesoderm, endoderm, and we are becoming an organism, like complete life. But if we talk about multipotent stem cell after the baby is born, starting from the umbilical cord to umbilical cord tissue to bone marrow to all our parts of the body cells, they are actually the multipotent stem cells, because they cannot differentiate into all three germ layers like ectoderm, endoderm, and mesoderm, but they have the ability to follow one particular lineage and give rise to many cells of that lineage. And now what you see here is the flow cytometry, which is very important for stem cell. And this is the advanced version of flow cytometry, which is VD FACS area. We also have other type of flow cytometries, which is responsible for the cell cycle analysis, cell differentiation, cluster of markers. Like we can see about a lot of CD markers, MSC markers. What are the different other immunology? Also, this is being used in characterizing the immunomarkers. So this is basically a CD marker characterization which can be done. But in FACS area, we can sort the stem cells according to different markers, according to different lineages. That is why it's called BD FACS area sorter. And this is how the marker binds. And what you're seeing is the differentiation. This is the adipocyte differentiation. And the last one is the osteogenic differentiation. So we are going to see how the stem cell differentiates. Now let's start with the basic thing of stem cells. If you see here, what happened before stem cell came? So stem cell aane ke pehle we have homeopathy going on, Ayurveda going on, allopathy going on, and then we have replacements therapy like organ transplantation, everything and anything. उसके बाद gene therapy भी आए, and then came the cell therapy, then the stem cells. So although there are various treatment options, stem cell आने के बाद completely it's taken a new world, I could say. That's why I welcome you all to this world of stem cells and regenerative medicine. So this image is very close to my heart. आपने इधर देख रही है कि the baby which is coming out of there. So it has two meaning. One is our life starts from stem cells. Like the embryonic stem cells is the one from the egg oocyte. After fertilization, the embryonic stem cells is the one giving rise to life. And second thing is stem cell has ability to break open and explore and treat many diseases in regenerative medicine. So, what is cells? As Swaranjit sir gave a great example, like there are trillions of cells in our body, and all the cells come from pre-existing cell, which is called omni-cellulare. Rudolf Virchow gave this term because everybody know that cell was discovered by Robert Koch. But how a cell gave rise to new cells, that's because of the mitosis, because of the regeneration ability that one cell undergoes differentiation, like division, and give rise to two different cells. This was given by Rudolf Virchow. Now, this is the complete picture about the discovery of cells slow and steadily. And all these discovery, why did I give this in this particular slide? Because all these discovery, the basic discovery of cells, gave us an understanding of what stem cells are. And without these important discoveries, we shouldn't have understood what stem cells are. Stem cells are from way early days as well, but हमको पता नहीं है. Like Prometheus was a person who was prisoned. And when he was prisoned, if you Google, there is a book called Regenerative Medicine. I forgot to add into this slide. उसमें क्या होता है? The person will be prisoned. Eagle को उसको liver खाने के लिए बोलते. Like when eagle come and eats his liver, उनको the king got surprised and Prometheus' liver got regenerated next day. But at that point of time, किसी को पता नहीं है कि क्या हुआ. But the truth is that it was since then later understood that liver got regenerated, and that is why his liver again formed. And since then, since Prometheus' time, the stem cells are there, and slowly and steadily they have started exploring and discovering. As Shruti said very rightly, and as Swaranjit sir also gave a great introduction about stem cells, they are unspecialized, undifferentiated, self-renewing. They keep dividing and becoming more stem cells, but upon physiological conditions, they have the ability to differentiate into any type of cell. So this is the entire way how the stem cell works within our body. Like the stem cell keeps renewing them, because one stem cell becomes two, two becomes four, as a normal mitosis process within the niche. But when there is a signal, when there is a need, for example, RBC, the lifespan of RBC is 120 days. So every 120 days, the stem cell, the hematopoietic stem cell has to give rise to new RBC. Similarly, lifespan of WBC is 16 days. So the hematopoietic stem cells within the bone marrow will give rise to new stem cell, new WBCs every 16 days. So how is it possible? These stem cells, the hematopoietic stem cells, which are actually present in the bone marrow niche, give rise to these hematopoietic progenitor cells which proliferate and then they differentiate into a mature RBC, WBC, platelets, NK cells, dendritic cells. All these hemopoiesis formation comes. The basic first thing is stem cells. Because way from beginning we've been reading about hematopoiesis or hemopoiesis in immunology, but the base of this hemopoiesis is the stems. This is what I've explained. This is from the cell. So here you can see how a stem cell differentiates into a progenitor cell. Here is a lymphoid progenitor and myeloid progenitor. And then slowly it becomes an immature reticulocyte, then mature erythrocytes. Similarly, it becomes an immature megakaryocytes, then differentiates into platelets. Similarly, the myeloid progenitor cells give rise to monocytes and the large monocytes. And this is how you can actually see the complete structure of differentiation, how even the basophil, eosinophil, neutrophil, B lymphocytes, T lymphocytes are all differentiated. So the base is hematopoietic stem cells. This is one very important example for the beginners to understand what stem cell actually does in our body. And this is a complete picture of how a zygote differentiates into inner cell mass and trophectoderm, and then what is happening in our body, how we get into the three germ layers, and how the placenta is also helping us and favoring us to form the three germ layers, and that's how the life starts. And that's where the stem cell begins. And as I told you right now, जब fertilization होता है तब से everything starts. As you know, immediately after fertilization, we know the cleavage. So this is the 2 cell stage, and then here comes the 4 cell stage, and then here comes the 8 cell stage, and finally it becomes like morula, then blastula, 32 cell stage, then 64, then 120. At this point of time, the inner cell mass, it becomes the blastocyst stage of the embryo, this red one, and the outer part becomes a placenta. The trophectoderm becomes a placenta, and the gap which you see here, this gap is called blastocoele, which becomes the amniotic fluid. And you can very well see this particular picture that this is the amnion, which is acting as a cushion, the amniotic fluid, the yolk sac, the umbilical cord, which is connecting the placenta and the fetus to the mother, and this is the placental layer. This is how it happens. And all of these has stem cells, and everyone has its own role. And that is why immediately after the delivery, as very rightly talked about, like Shruti said about cryopreservation, what they do is they take the umbilical cord, and this particular umbilical cord is being processed. They isolate the mononuclear cells, they characterize, and they cryopreserve in the exact manner how she explained. And then they can use it for research or they can use it later for any diseases. Similarly, the placenta, the amnion, they are also being recruited because it's just a trash. So we always say it's trash to treasure. So this placenta, which is a trash, the umbilical cord is of no use after the baby is born. It's just going to be a trash. Now from that trash, we are getting those treasures. And not only embryo, we also have adult stem cells. And adult stem cells also have huge potential. We are going to discuss about it. And these are some of the differences between embryonic stem cells and adult stem cells. The most promising field of embryonic stem cells are in vitro fertilization and nuclear transfer. But nuclear transfer, using nuclear transfer, you all know cloning, Dolly, you would have heard much. But human cloning is being banned because a lot of issues came because of cloning. And you all also know that embryonic stem cell has a lot of disadvantages, like they have ethical issues, they have teratogenic formation, and they cannot be induced into the adult because of carcinogenic activity. And for which Shinya Yamanaka in the year 2013, he discovered the great cells called induced pluripotent stem cells, which is from adult fibroblasts, the skin cells. They can become the embryonic stem cell like cells and differentiate into all three germ layers. So induced pluripotent stem cells, though we are not doing much in India, of course in Bangalore, NCBS, and in some of the places in Mumbai, they are working on iPSCs in a very crucial manner. Even in CMC, they are working on iPSCs. One of the lab is working on iPSCs, but not much explored yet in other labs. But it has got huge potentials to differentiate and give rise to many cells of all three germ layers and treat the diseases. This is one breakthrough we have. And of course, in adult stem cells, nowadays bone marrow adipose is being used, but recent advantage that we are going to see other stem cells. I'm going to show you pictures of isolation. Also, you can understand how other stem cells are also being explored and how the embryonic stem cells are being differentiated. They become embryoid bodies and then they start differentiating. This is one advantages of embryonic stem cell. When you remove the leukemia inhibitory factor, they become embryoid bodies. It is called spontaneous differentiation, one of the important factor of embryonic stem cells. Now, how the stem cells are classified. This is the basic understanding of stem cells. Now, when you talk about potency, as I told you, totipotent, zygote, the complete life. Embryonic stem cells. Multipotent. All adult stem cells are multipotent, which has its specific lineage. Oligopotent, few, like mammary stem cells. They are oligopotent stem cells. Hematopoietic stem cells, they are oligopotent stem cells. Unipotent and bipotent. If you talk about liver stem cells, initially the liver stem cells, which are differentiating into biliary epithelium and oval cells, which is hepatocytes. But later, they lose the ability to differentiate into biliary epithelium. The liver stem cells only differentiates into the important cell, which is called the hepatocytes. And that is why for people who are alcoholic, the liver keep on regenerating and they don't understand the importance, the impact of they are spoiling their own liver, kidney, heart, and brain because they get regenerated. And once a particular saturation goes off, they stop regenerating. The stem cells loses its potency to regenerate, or it would have affected the biliary epithelium as well. And that is where people turn the liver cirrhosis, liver fibrosis, and liver carcinoma. Based on sources, embryonic, fetal, and adult, most promising today in recent times, especially in India for clinical trials, are adult stem cells. And based on lineage, we have hematopoietic stem cells. We just saw mesenchymal stem cells, which are giving rise to mesodermal lineages like bone, cartilage. It is also giving rise to heart cells. It's also giving rise to neurons. It's also giving rise to the heart cardiomyocytes, the muscle cells, and so on and so forth. And there's something called site population. I'm naming it right now, and endothelial lineage and epithelial stem cells, which are giving rise to luminal and other epithelium. So this is how the complete differentiation takes place. And this is the definition which I gave you. There's one thing called unipotent. What is unipotent? As I told you, the liver stem cell is bipotent because initially in embryo it is giving rise to biliary epithelium and the hepatocytes. But later, the oval cell gives rise to only hepatocytes, and that's why it's unipotent. And nullipotent is a very important term. Why most of the people die out of brain disease or heart attack is because the brain and heart doesn't have the functional stem cells. It is just in quiescent stage, and once these cells degenerate, they don't have the ability to regenerate. That's why the heart cells never die. They cannot regenerate, neither the brain cells, and that's why treating the brain and heart disease becomes very difficult. But upon induction, upon some sort of inducement, definitely those cardiac stem cells and the neural stem cells can be induced to differentiate into that particular type of cell. But in human, in general, our brain cells and heart cells do not have the functional stem cells as they are nullipotent, which means they are in quiescent stage. And this is how the growth happens. Ethical issues in stem cell research are so much even till today, but it's better than before. Why am I going to talk more about adult stem cells? Because it's easy to isolate. There's no ethical concern, less invasive or non-invasive. I'm going to tell why I say adult stem cells are non-invasive, and it's highly plastic, which means it can differentiate into any type of cell. And these are the different uses right now people are using for adult stem cells in clinical trials and regenerative medicine. But most important use is they are using it as a therapeutic tool. And these are adult stem cells, which we spoke about it. Why adult stem cells also we spoke. These are the potential benefits. I again listed so that it gets registered in your mind. Please remember the 4 R's when it comes to adult stem cells. It's very easy to remember the replace, repair, regenerate, and rejuvenate. The stem cells alone in our body, amongst the trillions of differentiated cells, have the ability to replace, repair, regenerate, and rejuvenate. And that is why, as Deshmukh sir rightly said, stem cells are gold standard. Like in recent times, it's going to be a boom. It's going to be the future of tomorrow. And all you students have high potentials. That's because of all these 4 R's that stem cells can do. These are the advantages of adult stem cells as compared to embryonic and fetal, and that's why people are using it. So you can see here, this is overall the regenerative medicines. It's able to differentiate into all types of cells, especially mesenchymal stem cells have this ability because most of us have the problems of like muscle issues or cardiomyocytes, cartilage, bone, tendons, ligaments, adipocytes, and so on and so forth. And there is something called stem cell niche, where the stem cell lives. It's basically the home of stem cells, and it's going to stay quiescent there. And when the cells, differentiated cells, starts dying, it just comes out of its home and differentiate into that new cells. And these are the three major sources of stem cells which started from beginning for study. But not limited to this, we are going to explore different other sources as well, apart from peripheral blood, bone marrow, and cord blood. Now we will talk a little about all of these stem cells so you get interest into this field. This is how the bone marrow stem cell is being taken, the iliac crest region. So they just use the Jamshedi needle, and what they basically do is this: they just make the patient sleep so, and they just use this Jamshedi needle to ensure that it just goes to the bone marrow blood, and then they remove this particular blood, and then we are going to process it, and the mononuclear cells will be differentiated, and it will be isolated, characterized, and given to the patients for certain therapy. Within our body, what is its role? Very simple. This is the region where within our body यही बोन मैरो स्टेम सेल है जो हमको हेमाटोपोइटिक स्टेम सेल्स के द्वारा सारे ब्लड सेल्स को दे रही है, राइट? एंड mesenchymal stem cells के द्वारा सारे mesodermal differentiation. So this particular bone marrow stem cell is giving rise to all our blood cells, RBC, WBC, platelets to NK cells, dendritic cells to all the cells are differentiated, and the mesenchymal stem cells in the bone marrow are giving rise to bone, tendon, ligament, cartilage, muscle, and all of these cells within our body. Right now, I'm talking and you all are listening. Even right now, some of our bone marrow stem cells are differentiating in any of the cells which are less within our body without even our knowledge. This is how the process. So the first image, what you're seeing is the bone marrow which is collected. This is being processed in our lab when I was working in an institute, and it is being given for the patients. And this is the live image of the process which we have used. And then we use something called lymphocyte separation medium or the ficol, which is being added. So ficol ke upar in slide you have to just pour slowly and steadily these bone marrow blood. So they form two layers. And then you have to centrifuge it. Yes, we use a density gradient centrifugation method in a cooling centrifuge, and you have these four layers. The outer layer that is a plasma, and that will have all those plasma particles we separated. Then you have this middle buffy coat layer. You can see here this buffy coat layer, the second layer, is the mononuclear isolation, and the white one is the ficol or lymphocyte separation medium, and the last is RPC. Why this pattern? As we all know, higher the gradient, density gradient, lower the settlement. That's why RBC settles first and plasma settles in the top. Then we do a process of removal of the plasma. We took only the mononuclear cells. We do some process of washing. We do some process of RBC lysis, and then we get this particular pellet, which is MHC. And when you culture it, before you culture it, they are heterogeneous. They have blood cells, they have hematopoietic cells, they have fibroblasts, they have adipocytes, they have mesenchymal cells also, they have epithelial, they have endothelial. जब culture करेंगे, it just settles and that becomes a homogeneous culture. And this is the mesenchymal cells. Recently, in many decades, like at least a decade before or onwards, they've started using adipose tissue stem cells for therapy. Why? Because adipose tissue, there's a lot of procedures, abdominoplasty, liposuction, and these procedures they are using for reduction of weight. So these adipose tissues are just very easy in laparoscopic method to take. We know that we have two types of adipose tissue, omentum fat and subcutaneous fat. So we take that particular fat from the patient, we wash it, we mince it using some enzymes. It could be trypsin, it could be collagenase 4, it could be the other enzymes based on the condition of optimization in your lab. You would use that particular enzyme for digestion. After we mince, we digest it, and then we keep it in an incubator in an agitator. It keeps removing the cells. Then when you centrifuge, you can see the proper adipocyte, the blood, and the stromal vascular fraction, which is collected as pellet. Again, we take the stromal vascular fraction, we wash it, we use it with two, three other protocol steps would be there, and then we culture it and we grow those mesenchymal stem cells from the stromal vascular fraction, which is a heterogeneous population isolated from adipose tissue. Now, what the therapeutic application is? There's a lot of controversies when it comes to whether we use bone marrow or adipose. So people are still working on what is a better stem cell source and how do you decide? You need abundant quantity. It should be accessible. It should be non-invasive or less invasive for patients. It should be pluripotent, means it could be differentiating into neurons. It should have the potential to differentiate into heart cells. It should have the potential to differentiate into hepatocytes and many more other cells. एंड नॉट ओनली ऑटोलॉगस, मतलब हमारा बॉडी से हम खुद लेके पेशेंट को दे रहे, दैट इज ऑटोलॉगस स्टेम सेल्स. एनी adipose, which is any abdominoplasty which is there, or any bariatric surgery is being done, and that adipose tissue people just discard. वो डिस्कार्ड से लेके दूसरी पेशेंट को दे रहे, दैट इज एलोजेनिक. And adipose cells have been proven to be more allogenic as compared to bone marrow for simple reasons. Bone marrow में we have talk about HLA typing, human leukocyte antigen typing. So bone marrow allogenic देने में बहुत तकलीफ होता है and immunorejections आती है. So what people thought is adipose में more of mesenchymal population is there and less of blood, so allogenic के लिए हो सकती है, and that's how people started exploring adipose. And you have to retain the characteristics of all these three germ layers, and they are free from ethical issues. थोड़ी दिनों के बाद they started exploring stem cells from all sources. So bone marrow हुआ, cord blood, adipose. But otherwise also they started exploring stem cells from placenta, from menstrual blood, from endometrial tissues, from fallopian tube, from breast tissues and breast milk. And why these all additional sources? जितनी maximum stem cell potency explore कर सके and use for treating various diseases, people started exploring a lot of its applications towards regenerative medicine. And this now has gained a lot of insights because, let me show you here. This is the exact umbilical cord and the placenta you're seeing. This is the real picture. जैसे operation theater में after the baby is born, the cord and the placenta is removed, और उसके बाद वो cord को cut करके we took it and cord blood, that arteries and veins के द्वारा syringe से हम cord blood लेते, and after the entire cord blood is removed, you can see here the cord tissue. और इसको there are two types of stem cells in cord tissue. एक है directly tissues को ले लो आप, टुकड़ा टुकड़ा कट कीजिए, arteries veins remove करना है, not with arteries and veins, you don't have any arteries and veins, it's completely removed. फिर उसको टुकड़ा टुकड़ा करके explant culture करके put in media, and then from the tissues the cell starts coming up. That is cord tissue stem cells. There's another stem cells within the cord tissue जिसको cord tissue को open करें, that's called Wharton's jelly. It's a jelly like substance, mucus. This was discovered by Wharton, so that's called Wharton's jelly. उसको हम scrape करें, जैसे during corona cases, like that saliva scrape करके लिया, ऐसे scrape करें, collect those Wharton's jelly and process. So we have cord blood stem cells, we have cord tissue stem cells, we have Wharton's jelly stem cells, and we also have chorionic and amniotic placental stem cells. On all of these, we have published paper. If you wanted to know more, just go to Google Scholar, my ID, Dr. Indumathi Somasundram, and you can see my Google Scholar, the papers which we have published on all of these sources, just for your knowledge. Now, why is all this important? क्यों ये सारी stem cell research को हम करना है? Because as a student, many of you might get this question कि मैम ये करना भी क्यों है? Because tissue transplant मुश्किल है, organ transplant मुश्किल है, right? Rejections आते हैं, gene therapy भी बहुत मुश्किल है because you have to be very specific, otherwise it will also target other genes. इसीलिए cell transplant becomes easy, but cells are differentiated, so stem cell transplant becomes much easier. और दूसरी एक other issue है कि these stem cells अभी birth defects बहुत सारा आ रही है, genital diseases, hematological malignancies, उस conditions के लिए if a person, if a baby who is within the mother's womb is known कि उनको जरा जरा कुछ genetic issues है, उनकी goblet को लेके, like thalassemia, सिकल सेल एनीमिया, थैलासीमिया, सिकल सेल एनीमिया को पहले इतना सारा बहुत सारा ट्रीटमेंट्स नहीं था, बट नाउ व्हाट इज एडवांटेज, कॉर्ड ब्लड प्रिजर्व करके उस कॉर्ड ब्लड को एम्स, हमारा क्योंकि इंडिया में एम्स एंड अदर लैब्स हैव डन दिस, दे हैव ट्रांसप्लांटेड द कॉर्ड ब्लड स्टेम सेल्स इनटू दैट पर्टिकुलर पेशेंट, और पहला जो चाइल्ड थी उसकी कॉर्ड ब्लड लेके दूसरी चाइल्ड को ट्रीटमेंट देके थैलासीमिया की क्योर की. ऑल ऑफ दीस बर्थ डिफेक्ट्स आर क्योर्ड. Stem cell therapy for cancer is now still in clinical trial. It's not proven, but degenerative diseases like Parkinson's, cerebral palsy, Alzheimer's, spinal cord injuries, all of these are being treated by cancers. And not only that, stem cells को एक model organism तरीका हम use करें, because वो stem cell differentiate कर दे. For example, a lab, say for example, every student have come out with some drug which can treat your neurodegenerative or heart, कुछ भी हो. आप stem cells ले लो, stem cell को cardiomyocytes differentiate कर लो, and just put that drug in lab in vitro condition में, cardiomyocytes में वो drug add करके see the effect, whether the cardiomyocyte shrinks, it grows, heartbeats grow हो रही है, neurons shrink हो रही है neural के लिए, जो भी drug हो, stem cell acts as a model organism. जैसे कि Ambrosophilia है, we have zebrafish, we have this mouse model, we have lot many rat model, lot many other models, right? Monkey model, like that. We can use stem cell as a model organism to test the cytotoxic effect of any drug that is possible. So ये बहुत सारा differentiations होता है and there is a niche for it. And these are all the potential uses. ये सबके लिए अभी treatment है क्या? बिल्कुल नहीं. I'm not giving any false promise, but हो रहे. People are still exploring, trying and understanding कि जिस diseases के लिए हम stem cells use कर सके. And definitely in next 5 to 10 years this is going to be really a boom, right? अभी culture कैसे होता है? Just to induce the curiosity of all the students here, bone marrow mesenchymal stem cells आप देख लीजिए, subcutaneous की mesenchymal stem cells देख लीजिए और momentum fat का. See the morphological difference. All are mesenchymal stem cells based on the source. And Shruti excellently spoke about cryopreservation. अब एक बहुत जरूरी चीज है कि cryopreserve करने के बाद how long the stem cells will be alive, viable, उसके लिए हम कल्चर करना. सो व्हाट वी स्टार्टेड इज क्रायो प्रिजर्व करके फिर उसको लिया, क्रायो प्रिजर्व करके फिर उसको लिया, लेट पसाज तक हम कल्चर किया. तो आफ्टर 20 पसाज दिस इज हाउ द सेल्स डिफरेंशिएट और स्ट्रक्चरली एंड न्यूमेरिकली चेंजेस ना हो उसके लिए कैरियोटाइपिंग की, ऑस्टियोब्लास्ट डिफरेंशिएशन किया बिकॉज थ्री बेसिक डिफरेंशिएशन ऑस्टियोब्लास्ट, एडिपोसाइट एंड कॉन्ड्रोजन. So this is by oncocyte staining. Again, we confirmed osteoblast differentiation by alizarin and adipocyte differentiation by oilorate. And then not only its lineage, क्योंकि mesenchymal stem cells है तो mesoderm देते ही है. Trans differentiation. Mesenchymal stem cells endoderm दे सकते क्या, ectoderm दे सकते क्या. So these are beta cells. We converted the stem cells into beta cells also. And this is flow cytometry, BD fax area. The sorter, इसमें से हम cell sort भी कर सकते. Right. These are the advantages also. And we have done the therapeutic application needs. So हम अभी क्या किया? Initially with my senior, he is my senior with which lab I worked. With him, I was working on bone marrow, adipose, cord blood. But I wanted to do something beyond. So I chose my PhD in 2009. I registered for my PhD. In 2009, I wanted to explore. First, I started my work 1 year with cord tissue. Then all of a sudden, I wanted to work on endometry. So I requested my guide, I requested the university to change of the topic, but I promised them to finish on time. And after that, I changed my source into endometrium. These are the three sources which I took in my PhD: endometrium, fallopian tube, breast milk. क्योंकि जैसे जैसे मैं study किया, I found that endometrium, which is the student's cell, has immense potency to differentiate, because within our body every month we have this, right? It goes like the regeneration. After every menstrual cycle, the endometrium regenerates. 400 cycles of regeneration undergoes. And these are the reasons why I study. It's got two different things. It has got vascular regeneration, high for angiogenesis, and the problem is it is also having some gynecological problems. So we started exploring all these endometrial stem cells. We will be finishing in last 5 minutes. I just please give me another 5 minutes. Why endometrial stem cells? Because of embryological origin. Every woman, the child who is born, the female child, angiogenesis starts in utero. This is how the angiogenesis starts. It has got dynamic cyclical changes, tissue remodeling happens, and what happens is endometrium is just a waste. Many hysterectomy cases happens, uterine prolapse, uterine fibroid. So many hysterectomy cases happens, and they just scrape the endometrium. What we do is after a case of hysterectomy, which is uterus removal surgery, we take the endometrium, cut open it, and then scrape the endometrium, and it is completely allogenic and it can store long. We have worked till Passage 20 for endometrium. We have cryopreserved and it is working. This is how the three different uses of endometrial stem cells. And what I proposed in my study is, and we after that, after I completed my PhD, I extended the study to my own PhD students. Are this endometrial stem cells can treat any diseases which is respectively to angiogenesis. बोन मैरो ब्लड रिलेटेड डिसीज के लिए यूज हो सकते, एडिपोज अदर डिजनरेटिव के लिए यूज कर सकते. एंड वी प्रपोज दैट endometrial stem cell can be a great source of treating infertility, and not only that, it can treat ischemia, it can treat cardiovascular diseases because it has high angiogenesis potential. And why breast milk? As you see, a drop of the good stuff. Every mother's milk has a lot of immunofactors, water, proteins, hormones, cytokines, and recently they found that this particular mother's milk has highly enriched with the stem cells. So we did a small experiment using chick embryo, the angiogenic study using endometrial and breast milk, and we found that स्टेम सेल थेरेपी कर सकते, targeted drug therapy also हम कर सकते for gynecological disorders. This is one such disease called endometrial hyperplasia. You can see here, which is turning to endometrial cancer. So इस stem cell को हम target भी कर सकते. So endometrial stem cells are double-edged sword. On one way it is positive, it can treat any diseases. Other side it is mutation, it causes endometrial hyperplasia and cancer, where those stem cells can be targeted. So these are the basic study. I've written a basic book called Stem Cell Biology Practical Manual. Unfortunately, I've not done much marketing. Abhi, I'll be happy to share this book with very low all the students if you wish to know more about stem cells. It's available in Amazon. But personally, I'll try to give my few copies to you with very low cost. And finally, the last slides. What are we doing for the students? So initially, I was into full-time job, but I don't want to restrict my knowledge to just that particular population. So we started giving a lot of training programs. And as Deshmukh sir rightly said, Microbiology Society of India is always like very supportive. And now we are happily, I'm saying that we are going to explore in collaboration a lot of things for all life science students, hands-on training on many life science techniques, workshops, webinars, internship, final year projects, and not that. बहुत सारे स्टूडेंट्स को ये प्रॉब्लम आती है कि मुझे इंग्लिश नहीं आता. So I thought all life science students should also develop that skill. So we started spoken English one-to-one communication, placement trainings, one-to-one career guidance, and all life skills. Life skills matlab art of reading, writing, speaking, communication, listening, focus, mindset, how we need to have a good mindset, growth mindset, how do you manage your stress, how do you embrace the challenges. से बहुत सारा चीज़ों है because in so much of fast pacing world we have a lot of issues, and most importantly we also give the career guidance and goal setting and mastery for the students. And with this I conclude my talk. There are a lot many things to go. Stem cell for students, I wanted to highlight just 1 minute. You can be a stem cell faculty in academics, get into research, be a technician, or be a researcher in any stem cell cord blood banking or human milk banking. You can also get into a lot of publication sectors where they welcome many stem cell sources. You can work in different national and international laboratories, research labs, institutes, and pharma companies also, like biotech companies and pharma companies, and enhance this particular stem cell knowledge. A lot, many things are there to explore and to get into job. The only problem is students are not aware about what are the fields that they can explore and get into the jobs in STEM cells. So please do pay attention, take that curiosity and drive, and explore what you can do best in STEM cell and get your placements there. And these are my contacts for your reference. So all the very best, and thank you so much for your kind time and attention. Thank you, sir.
Thank you
Wonderful
very much.
presentation, ma'am. We enjoyed a lot, and we are very grateful to you for your such an informative lecture.
I
Very,
think
very
I was
grateful.
on time. Did
And
I exceed time? I'm so sorry if that happened.
very timely, very accurate, man. By all means, audio, video, everything was clear, everything was perfectly all right. Thank you so much, ma'am. And now I would request to the participants, yeah, please go ahead with your feedback, with your comments. Along with that, the Q&A session, the question answer session, I would request the senior participants to please and our guest of honor, Dr. Suranjit Singh sir, our special guest, our chief guest, sir. Please, sir, you over to you all.
Ma'am, may I ask you a
If you
question?
Yes, Donna, please go ahead.
Yes, ma'am. As you were talking about that breast milk, so recently I came up with an article that microplastic is getting into the breast milk. That in some researches they found that there is microplastic in the breast milk. So how is it getting into breast milk? Like we are using plastic for lots of years back. So why it is coming recently into the breast milk? Because it's injurious to the health of the kid which is consuming that breast milk. So what can we do to prevent that things?
Very, very thoughtful and very good question. Actually, I'm so glad that you are aware about microplastics. You know what? Recently, we organized along with the Mulai Academia, TISIL, and Microbiology Society an offline workshop on health, World Health Day. There was one talk on microplastics because everybody know that use of plastics is harmful, this that, but we skip these minute microplastics which we consume in our day-to-day life. It would have been before itself, Duna, but it is not brought into the notice. Somebody like, now recently the microplastics are gaining importance. In day to day life, we are using a lot of microplastics, and these microplastics are slowly and steadily getting into our body, which has come to attention. So, one particular group or several other groups, they thought, why don't we see if at all there are microplastics in breast milk and is it harmful? And they found yes, but there are some controversial thought also, because it is still not proven in detail how, why, and what way the microplastics are present. So I could say, of course, what you say is right. Microplastics could be in breast milk, and yes, after infusion, it could be placing a cause in child's body. But these microplastics do not create harm immediately. That is the biggest problem. The microplastic slow and steadily gets into our body, and it just like get tagged slowly and steadily. So what we can do? Nothing can be done as of now. Individually, what we have to take precaution is that we have to maximum avoid these microplastics. Like, say, very simple example is whenever we go, we buy, even in meetings, we have these plastic bottles. So people feel it's just a bottle and we're just drinking it and we're throwing off. No, the entire plastic bottle, as in when we, drink water so many times, those microplastics, which is within the bottle, gets into our body. We drink water through plastic. We eat through those plastic things. So we are not aware about those things, and that's how it gets into the body. And what happens? Very simple. The blood takes it to everywhere, and that's how it's also gone to the breast milk. And people have found that there are microplastics. It's harmful. So what can be done?
Ma'am,
Yes, it's just an awareness, and we have to be very cautious, and we have to read more about it, understand all possible ways of avoiding plastics and microplastics. And this that study is there too.
okay, thank you, ma'am.
Any other questions?
Just some curiosity.
Yes, sir.
Just a curiosity. Um, you mentioned about gene therapy, and then uh, stem therapy seems to have come as an advancement over the previous one.
Yes, sir.
Uh, it appears that this system of treatment is being applied so far only for treatment of different types of cancer.
Sorry, sir. Different types of
Cancer.
cancer. No, sir. You mean stem cell therapy?
Yes, stem cell therapy. Is it being applied for
No, no,
treatment
no,
of
sir.
diseases other than cancer?
Yes, sir, I'll tell you. Actually, the stem cell therapy for cancer is just now started getting its importance. So initially, the stem cell therapy was being treated for the genetic diseases like thalassemia and sickle cell anemia, and then they also started treating for liver cirrhosis, cerebral palsy, spinal cord injury. So there's a, if you get into this particular clinicaltrial.com, so there are stem cell clinical trials which are registered in the government sector. So there are various stem cells which are being treated for hemophilia, thalassemia, sickle cell anemia, cerebral palsy, Parkinson's. They are using stem cell therapy. Liver cirrhosis, they are using stem cell therapy. Spinal cord injury and liver cirrhosis, most commonly they use for stem cell. There is a few studies where even for cleft lip palate they used some stem cells. And while treating some of the cardiovascular diseases, what they do is we use a stent therapy, right? So within the stent, they infuse these bone marrow cells, assuming that those bone marrow cells would localize home into the heart, and then they would differentiate. They help those cardiac stem cells to differentiate into cardiomyocytes. So they have been used for these such therapies. So these were actually potentially being used from beginning. Later, once they thought that initially what happened was during these times, the cancer was only treated using chemotherapy, radiotherapy, immunotherapy, surgeries, and all of these. But then the cancer used to relapse. Again, they used to give chemotherapy, radiotherapy, surgery. Cancer used to relapse. And then they started NK cell therapy and interferon therapy and immunotherapy. Still, cancer used to relapse. And then we found that there is something called cancer stem cells, where this is the niche. And then from this cancer stem cells, there are many cancer cells which are growing. Now, all the treatments are only at the superficial level. And after this is killed, this is again giving rise to more cells. So, recently, the stem cell therapy for cancers has been most important, especially for leukemia and other myeloma related cancers. They are trying this. So, what, as you rightly said, stem cell therapy are given for cancer patients, but before that, all of these other treatments are also very promising.
Okay. One more uh, thought came.
Yes,
You
sir.
also mentioned the ethical issues.
Yes,
So,
sir.
um, what happens actually? Does it harm uh, the baby or the embryo from whom
I'll
the
tell you. Yeah, I'll
the
answer this. Sir, in India, basically embryonic stem cells is banned. So we don't work on embryonic stem cells at all. If at all people are using those in vitro fertilization embryos, the excess embryos which are used for IVF labs, they have that ethical concerns and they have to clearly inform the government and ethical clearance that we are taking these embryos from the in vitro fertilization lab only for research and education purpose, and this is not used for trial. So most of the stem cell therapy which are going on in India and even in most of the other countries also, they work on adult stem cells.
oh,
So when it comes to adult stem cells, they are only working on stem cells like bone marrow, cord blood, adipose tissue widely, and there are few fertility labs and other labs where they are using these endometrial stem cells for the increasing the pregnancy rates and treating infertility for thin endometrial patients and for the follicular studies they are using. So this is completely different. So there's no question of getting harmed with embryo. Earlier, there was one study, sir, where the in vitro fertilization labs used to grow many embryos, and illegally they used to give it for stem cell research. And some of them got caught, some of the labs got closed, and all these were unethical. But then there was a stringent rule that if at all any IVF lab they are taking the embryos, mostly not. If at all we are working on embryos, then we need to definitely give a consent from the patient, from the lab, and to the particular authority of ethical committee without signing this consent saying that we are taking only for education or research purpose. The embryo should not be used for any stem cell studies or it should be discarded. Directly, the embryo should be discarded.
thank you, thank you so much.
I have a
Thank you,
question
Dr.
in
Indumathi
chat
ji.
box.
My question,
Yes, sir.
you have delivered a very good lecture, and very informative in a very simple language. So thanks to you. And as far as India is concerned,
Sir, I'm not able
it
to
is
hear you.
always be competitive in the sense that in missiles field, in space
Sorry, sir.
technology, we too stand. So you tell me that where
Sir,
India
I'm not
stand.
able to hear you.
Okay, and are you? Am I audible now? Am I audible? Am I
Yes, sir.
audible
Yes,
now?
yes, sir. You are. You are.
Yeah.
Please.
So my
Yes, sir.
question is,
You are.
thank you very much for your very informative and lecture regarding the stem cell treatment. Where India stand as far as the stem cell therapy is concerned? First question, as compared to the
Yeah.
US and other advanced Japan and other countries.
Hmm.
Second question is that
Hmm.
most of the sports injuries, and especially it happens with the those who play football, hockey, etc. So after, if it is injury is there,
Yes, okay.
so it remains for a long time. The patient goes, and it takes a lot of time. So can this therapy be used for knee replacement or cartilage damage? So over
Yes,
these
sir.
two questions, I would like your
Yeah, I have
opinion.
noted the question.
Yes.
Yes, sir. Yes, sir. I'll first tell you about the first question I'll answer. Where exactly does like India is in reference to stem cell? To be very honest, we are not very competitive as compared to other countries who are working on stem cells. That is for sure. But we are trying our best. Especially in India, there are many treatments where the stem cell therapy is being given is the degenerative diseases, spinal cord injury, and bone and arthritis problems. So the people are working completely, as you said, first question and your second question's answer is the same. In India, people are working on three fields, most prominently working on stem cell therapy for degenerative disease, stem cell therapy for fertility related diseases, and in those degeneration also for arthritis, osteoporosis, there are some positive results where the stem cells, especially adipose, as compared to bone marrow, are being used, and people are using this for stem cells to cartilage, stem cells to bone. This is very prominent. And also stem cells related to spinal cord injuries and other neurodegenerative diseases, people are exploring. And there are two labs who are eminently working on induced pluripotent stem cells also. So, in fertility also, there are many labs who are trying to use these stem cells and trying to increase the pregnancy rate, use these stem cells to ensure the follicles grow, like from these stem cells where they can differentiate into those follicles and all this. So, definitely, I could say when I was doing my PhD, as compared to that period, which is in 2009, 10, 11, and 12, as compared to that, now in the last 12 years after completion of my PhD, I can see a great advancement. And you can see after IPS also, there are great advancement. So the I could say that it's still a trial, but the percentage of improvement of diseases. In earlier days, if it is only 20-30% of improvement, in recent times, if there are at least 50 to 60% of improvement that has happened, that is because of the diverse approaches that people are trying to use in stem cell. Like some are using bone marrow for specific diseases of genetic disorders for other blood related diseases. Some are specifically only working on adipose tissue related to the differentiation of cartilage, bone, muscle. And as you asked about this sports person knee replacement, there are some tissue engineering studies and biomaterials which is combined with stem cells using 3D print. There, what we have started doing is they have started creating the organ. What they do, sir, they take the stem cell, they take those 3D printing method, complete decellularization of that particular organ from any source. Complete decellularization will be done, and then those will be given new cells, and they use a scaffold or any of the basic material, and upon that they grow those tissues, and that's how the ear pinna, that's how the cartilages have been grown, and they have been placed for human, like growing the cartilages, growing the actual ear pinna. So all of these with respect to tissue engineering, biomaterial, 3D printing, and stem cell put together, they are doing this. So definitely in next decade, if you see, the stem cell could be ultimate source of organ replacement. Even small organs can be replaced using stem cell is what I am looking for. So we will hope for the best.
Uh, thank you, Doctor Sahab. Thank you very much.
Thank you, sir. My pleasure, Umesh. I think Shri
Doctor
has raised
sir,
her hand.
Dr.
Uh,
Indumathi ma'am, just tell me
yes, sir.
what is how this stem cell technology treatment differs from allopathic treatment. Allopathic doctors are required to be trained in stem cells treatment also, and whether stem treatment technology experts are treated as are qualified like MBBS doctor.
Yes, I'll answer. Sir, allopathy doctors now are learning stem cells. They are, for example,
Yeah.
there are few doctors who are into, like they wanted to, like if there is a bone replacement, or if there is a sort of cartilage replacement study to be done and arthritis to be done. So they have started doing this. There's a group also who are exclusively working on these stem cells. So those specific doctors, specialized doctors, started learning stem cells, and they have started giving therapy. But according to me, if you ask me, in none of the allopathy doctors should actually individually carry these stem cells to the patient. There are some groups, like there are two school of thought. There are one group who are doctors with respect to other stem cell scientists, technologists, and their approval, the basic science and doctors together, they are working on some protocol and treating the patient, which is fair enough. But there are some people who are having a superficial knowledge of stem cells, but still they go study, get the degree, and they call themselves as stem cell specialists, and they alone work on these particular very minute, different, some even they give that plasma therapy. They have those PRP therapies. They also have the GCSF therapy done, and then they try different methods of working with patients on stem cells. So personally, if you ask me, sir, that is completely wrong, because allopathy doctors, it's good that they should get the interest, because with only the surgery, or with only the technological advancements, with drugs or with surgeries or with medicines, not all diseases can be cured. So allopathy doctors taking interest in the field of stem cells, working with stem cell researchers, working with stem cell technologists, and trying to do a better therapy for patients are very good. But it has to be with a standard protocol. They should come in unison. Clinicians and researchers should go hand in hand. Then, if they take it forward, I think we could get immense, humongous results. Which is right now, if you ask me, it is little lacking. It should be.
There is one
Clinicians and researchers should combine, go hand in hand, and start working for the patient.
I will
Now, when this MBBS doctor feels that here stem cells technology will be better than allopathic, so they are taking use of your people.
Yes, sir. Which is
They are
unethical. Yes.
they are
It
taking.
should be followed with a standard stringent protocol, and then they go to the patients. That would be more feasible, and it's more promising also. Like, lot many things could be done.
Hmm. Thank you.
Shaili, I think she left the meeting. Shaili was raising her hand, and Usha, your question?
Very nice talk and nice presentation.
Thank you.
Actually, when I was working in Chennai, I'm working under the guidance for STEM
Usha, I'm not able to hear you. Can you just please be little louder?
other than speaker, please unmute your mic.
Yes, ma'am. And it was a wonderful presentation, ma'am. When you're working in Chennai, it's working under your guidance. The stem cells fields when you're working on the water and jelly, cord blade, and all. I have worked. So
Oh, is
you're
it Usha Rani?
yes, yes, ma'am.
Oh, so nice to see you here. Very nice. Thank you, Usha. Usha
Still,
Rani was
yes,
my student. She was working with me when I was in the private lab, where I was working in stem cells in my first job in, I think, May 2009, right?
yes, yes, ma'am.
Oh, so nice to
2009.
see you here. Yeah, yeah, Usha, please go ahead.
Still, the craze is not settled down. The only thing is the cost is very high to deal with the samples.
Yes.
That is the only thing. We don't have fund at present. We are working on the reproductive toxicology, PCOS, endometriosis, pregnancy samples. Also, we are dealing with. Only thing is we don't have like funded. So so we didn't start that work stem cells part and all. Only we are dealing with the toxicology related. So
Excellent,
in
excellent.
future we can have a collaboration.
Yes, yes, please do contact me. And I'm so
Yes,
glad
yes.
to the other participants and the guests I'm telling. So Usha Rani was my,
Thank you.
she came
Into
from
into
very
one
advanced setting,
specific
right?
question.
Yeah. Thank you,
Dr.
Usha. Thank
Indu, one
Thank
specific question:
you.
whether thalassemia can be treated through, I mean, stem cell therapy of a 2.5-year-old child,
Sorry,
and
sir.
the thalassemia is
Just
hereditary.
repeating your question. Sorry, whether?
I repeat, a 2.5-year-old child
Ah,
has got thalassemia,
ah.
which is hereditary.
Hmm.
Thalassemia,
Yes, sir.
hereditary thalassemia can be
Yes, yes.
treated through a stem cell.
Yes, sir. It can be. There are cases also. I can present some case study later in the group. For thalassemia, there are many curable studies. Two possibilities are there, sir. One is for thalassemia, if the child is found to have thalassemia, and if they have any siblings cord blood which is stored, that can be used for a treatment. If not, the bone marrow transplant is the only cure. So what they can do is they can choose a suitable donor, and they can use these bone marrow transplant and use these stem cells which are produced in the bone marrow, and then transplant into this child, and there are cure cells. So either the cord blood or bone marrow, and there are many clinical trials, proven clinical trials in India also, where they have treated thalassemia with cord blood and bone marrow for newborn babies. They wait for like 2.5, 3 years based on that doctor decides, and thalassemia patients have been cured using cord blood or bone marrow. It's possible, sir. People are doing.
I
There are
had a,
many studies.
I just
Thank
want.
you.
I had a friend. Um, his his case was thalassemia major, and what I learned from him that every, I mean, repeatedly the bone marrow had to be administered.
Yes.
So is that what I'm talking about 20 years back.
So
Uh,
now it need not be the case where repeatedly, because there they've been doing bone marrow transplantation. So here they are injecting those stem cells. So probably what will happen is there is certain point of time based on severity that maybe 3 4 injections needs to be taken, but then not necessary for like how we used to do for insulin injections. Not necessary lifetime that person has to take these bone marrow courses.
I see. Thank you.
Thank you, sir. So we have a question in the chat box, Arpita Bhattacharya. So her question is, who can donate stem cells? Like, what must be matched between the donor and the receiver? That's a good question. So Arpita, we cannot actually donate a stem cell without a lot of. There's some ethical things, but still, if all those ethical issues are resolved, and I'll tell you what can be done. So recently, what people do is bone marrow transplant. The stem cells can be donated only with siblings or with any relatives where HLA match. So what is this HLA? Suppose I am there, we have this blood group. Even I'm O positive and you're O positive, or we have the same blood group. Giving blood to you and you taking the blood is different. But when I'm giving you the bone marrow transplant or when I'm giving you the stem cell, there's something called HLA, human leukocyte antigen, right? So HLA matching is very, very important. So when a person is sick, for example, I am sick and I wanted my own bone marrow to be transplanted, absolutely no issues. But suppose I'm sick and I want someone else's bone marrow to be transplanted, definitely one thing which they will ask for match is HLA type. There's something called HLA-DR, DP, and DQ, which is very, very important. HLA-2 typing. Unless and until the HLA matches, there cannot be any donation. And this HLA will match only with our own kith and kin, like parents, siblings, and very close relatives. Otherwise, that will be completely rejected. But there is another option apart from bone marrow or apart from match. Suppose if any person is undergoing hysterectomy, or any person is undergoing abdominoplasty where they wanted to remove fat, plastic surgery. They wanted to just be beauty, so they wanted to get rid of the fat. They can say that, I have heard that there are some chances where adipose tissues are being used for stem cells. I wanted to just give off that adipose tissue the lab can use for stem cells. That way they can donate. If any person who is undergoing hysterectomy, they can donate their, the endometrial stem cells for research or for study. Cord blood. There are many private labs who are using these cord blood store and then using for therapy. But if you wanted to donate, if somebody like says that, now if you ask me personally, that's completely, I'm raising a disclaimer. I'm not harming or naming anyone here. But if you ask me personally, if suppose there are many people who ask me, like, after a child is born, should we have to mandatorily go for cord blood stem cell bank? This is a very prominent question which everybody asks. And for me, if you ask me, I'll say, unless and until the child who is inside the mother's womb is found to have some genetic disease, is found to have some blood related diseases, or found to have some hereditary disorders, or hereditary wise, that particular family has got some issues, there's no need of storing that particular cord blood, because cord blood, which is supposed to have so much years of viability, actually is not. And if at all the child is grown, say after 10 years, the child could have some problem. After 15 years, the child could have some problem. There's always autologous bone marrow, which is there for giving the therapy. So what happens is the child's blood for no reason is being stored, and every year the parent is renewing. That has become a fantasy now. Every parent are doing, and it's been a marketable product. But if you ask me officially, my personal opinion is unless and until there is a need, the cord blood need not be preserved. Or within the family, if there's, like, for example, in thalassemia, I just answered Sir's question. The second child is found to have some issues, or hereditary, they have some issues, or a caution. And if there could be a reason, then the cord blood definitely mandatorily needs to be preserved, because that could be of use to any of them. So I hope I've answered the question. This is how the match comes into role. This is how the donation of stem cells comes into role. This is not like how we donate organs. So yes, thank you all.
Yes, ma'am. Thank you.
Thank you all.
Thank you.
So I think we are done with the session. Anything else is left?
Amitabh ji,
Today, means Mr. Chandreshwar Khan, who used to speak at the end of this of every webinar. So we cannot hear his Amrit Bani because he tried too much, but there was some disturbance in his telephone. So he rang me so many times during the webinar going on. No, so we can't listen him, but
Ajay sir is
we
there.
can.
Ajay sir is there. Ajay
अजय
sir
सर
is
इज
there.
देयर ओके दैट इज वेरी गुड अजय सर
Yes.
आप अपना ब्लेसिंग्स दें और
Yes, please, sir.
प्लीज कमेंट ऑन टुडेज वेबिनार इट वाज वेरी नाइस वेबिनार और बहुत ज्ञान हम लोगों ने इसे लिया एंड एज आवर वेबिनार इज फॉर शेयरिंग आवर नॉलेज एंड स्किल्स नो और बहुत सुंदर नॉलेज है कि स्टेम सेल्स में हम लोगों स्टेम सेल्स टेक्नोलॉजी पर हम लोगों ने जिस पे हम लोग जीरो थे एंड वी आर नाउ नोइंग वी हैव कम टू नो समथिंग नो तो ये ड्यू टू इंदुमती मैम डॉक्टर संजीत सिंह जी डॉक्टर देशमुख जी इन लोगों के उनसे हम लोगों ने बहुत कुछ सीखा सो वी वांट टू लिसन फ्रॉम अजय सर
Yes, sir. Yes,
अजय
Ajay sir, please,
श्रीवास्तव
sir.
साहब अजय श्रीवास्तव साहब
Dr. Indumathi, it was a terrific lecture. Like us,
Hello, Ajay sir. Good evening. Nice to have you here.
good evening, Indumathi. Yeah.
अमिताभ
We
सर
are,
म्यूट कर लीजिए,
yeah,
वॉइस इको कर रहे
हाँ
हैं, अमिताभ
हाँ
सर म्यूट कर लीजिए.
yeah. What I want to say is that, what I mean is that if in our society, out of 100 people, if half person was educated about stem cells after today's lecture and after listening to watching the recorded video, more than 10 to 20% out of 100 will be aware of this thing. So there are possibilities. You have explained it very well about the diseases that we normally used to condemn the person who has those diseases or that situation. Now there is a ray of hope for uh, the people. The sufferings will certainly come down. And through your lecture, you have also reinforced the idea that yes, the students can take up lines like this, and there is enough scope, and you can pursue your career in that. And we are together. We are just spreading happiness. This is the yardstick. This is the landmarkish thing that events like this are happening. I'm very thankful to Dr. Indumathi Somsundaram, Shruti Chavule, Dr. Amitabh sir, Nagesh Jha sir, A.M. Prasad sir, Sarat Subarpine, and everybody else whoever participated in this webinar. Thank you very much.
Thank you, sir. And tomorrow we are going to listen another great scholar, Dr. Swaranjit Singh. So tomorrow I will request all persons, no all participants to take part in tomorrow's webinar also. He has prepared his presentation very nicely and very coolly he used to say everything and narrate everything. So tomorrow we will again be lucky to listen such a great scholar, Dr. Swarnajit Singh जी हम लोग कल आपको सुनेंगे और हम अब चाहेंगे अपने माने हमारे जो चंद्रेश्वर खान साहब नहीं थे लेकिन हमारे प्रसाद साहब हैं वो सभी को धन्यवाद देते हुए और सबको आशीर्वाद अपना देते हुए सबको विदाई का ये कर दें या शरद जी ने कुछ और सोचा है तो शरद जी कैन डू दैट शरद
नहीं बिल्कुल सर महेश्वरी सर
हाँ महेश्वरी साहब हाँ महेश्वरी साहब आप जरा कुछ आशीर्वाद वचन देते हुए क्योंकि हमारे ग्रुप में सबसे बुजुर्ग आप हैं और आपके आशीर्वाद से ही हम लोगों का इतना सफल हो पा रहा है वेबिनार्स और हम लोग इतने दिनों से 2-3 साल से करवा रहे हैं और ये यूनिवर्सल वेबिनार हो गया है दुनिया के हर कोने से इसको लोग सुन रहे हैं और दुनिया से हर कोने के लोग इसमें बोलने के लिए और भाग लेने के लिए हमें रिक्वेस्ट करते हैं हम लोग बहुत लकी हैं कि ईश्वर की कृपा से इतना अच्छा एक वेबिनार ग्रुप, अनुभवी लोगों का ग्रुप तैयार हो गया, जिसमें सारे विद्वान हमारे अपनी अपने एक्सपर्ट नॉलेज को देते जा रहे हैं। हमारे बच्चों को बहुत लाभ होगा जो सुनेंगे। बच्चों को शरद जी, आपको, आप मीडिएटर हैं, तो हम लोग चाहेंगे कि शरद जी और बाकी जितने भी स्टूडेंट हैं वो मोटिवेट होकर वो एक दूसरे को मोटिवेट करके इसको सुनाएं। हम लोग इस उम्र में भी बहुत कुछ सीख रहे हैं, तो उनको तो बहुत लाभ होगा। उनको अभी काफ़ी जिंदगी जीनी है, एंड दे शुड ट्राई टू इम्प्लीमेंट वॉट एवर दे आर लर्निंग फ्रॉम these our webinars, no excellent webinars of every day, every Saturday and Sunday. So, Prasad साहब।
सर सॉरी टू इंटरप्ट, मैं कुछ बोल सकती हूँ? फॉर देयर इज
हाँ
अ
हाँ
क्वेश्चन फ्रॉम द
हाँ हाँ।
स्टूडेंट।
नहीं, इंदु
या
प्लीज पहले अपना फोन नंबर लिखा दीजिए, डॉक्टर इंदु प्लीज।
या सर, I will. There's one question. This is my WhatsApp number. Yeah, for all the students and others, there's one question in the group. How can we study more about stem cells? So you can contact me, and also there are informations online. But there are some books also. I have written personally the books, so I'll try and help you out with understanding different labs. We are starting with the Microbiology Society of India. We are starting a hands-on training program and course on stem cells shortly, which is I think without Deshmukh sir's permission, I have announced it here for. So we will definitely post it in all the groups. So stem cell के लिए जो भी करना है, even for cell biology, stem cell, molecular biology, bioinformatics, all life sciences, I would be really grateful to help the students because in my 15 years of career, जैसे the Human Rights Association is working, Jan Jagran is working. Recently, I came to know, but from very beginning, I'm very grateful to my Microbiology Society of India, Deshmukh sir, and the entire team. So they all have inspired me. So it's just a teamwork and the journey. Let us share our complete knowledge to the student community and uplift them, and let us also grow. So definitely we will help you all, and thank you. So there was a question I missed. That's why. Sorry, sir. Over to you. And thank you very much to Jan Jagran and Human Rights Association. Because without this initiative, especially Amitabh sir and Swaranjit sir, and even Sarat Chopra sir, Deshmukh sir was the pillar behind for whatever I am today. And also our chief guest, A.M. Prasad sir, and all our participants. I'm sorry if I've missed someone. Without you all, this webinar wouldn't have been a great success. And I feel so blessed and privileged to actually be here and present my little knowledge to the community, sir. So my heartfelt thanks to all of you for giving me this opportunity. And thank you, Shruti. It was great listening to you.
Are you conducting any certificate course on stem cell technology at present?
Yes, sir. We are starting. Yes, sir. We
You
are
are
going to
starting.
start. I will give you official notice, sir. Yes, we are going to
Acha.
start.
Of how many duration? How
So
much
we have a
duration?
certification program for 3 months and 6 months. And
Three
we also
months and
have
6 months.
and we also have internships and hands-on for 1 week to 10 days. Certification program for 3 months to 6 months.
And
Durgesh,
for that science background is required, or for that any commerce, arts, whatever
yes,
background
it's
may be
like
the
science background, sir. Any life
science,
science, any biology and science, any life science.
biology and science
Yeah, any life science.
background is essential for that.
Yes, sir.
Yeah, I want to know that. Okay. So can this means stem cells experts be better substitute of MBBS doctors running at present
Yes, sir. They
doing?
can also be a part. We are inviting experts. So we will be a team, and very soon we would be most probably before this month, and we would release that. Mostly
Thank
by
you.
March.
Very good, very good. Prasad Saab, Ashirwad
Durgesh,
Bachchan, Amrit Bani.
so Durgesh, you had a question, Durgesh ji.
Dr. Indu, I cannot jot down your phone number. It is 952-914 after
So 95299 51614.
952 double
299
nine
51614, sir.
51
614, sir. 614.
51614. Thank you. I
Yes, sir.
repeat, 952
Double nine
No, no, I am repeating. 9529951614.
Yes, sir. Yes.
Thank you. Thank you.
Yes, sir. And that's my WhatsApp number and call. But if you directly wanted to call, there's one number for my personal number for calling, which is 8788382033. That is only for calling, sir. Uh,
Now I will converse on WhatsApp only. Thank you.
yes,
God
yes.
bless you.
Then that is
Thank
the
you.
number. Yeah. Nowadays, everybody uses WhatsApp. Yeah, Durgesh, you had a question. I think then Prasad sir wanted to speak, so we'll just quickly finish your question and listen from Prasad ji. Prasad sir, can I, with your permission, answer Durgesh question alone? I know it's time. Yeah.
Yes,
Durgesh ji,
sir.
please.
Yes, ma'am.
You had some question?
No, ma'am, not immediately.
Okay.
Prasad sir, please go ahead.
Prasad Saab.
I think I have unmuted, yes. Dr. Indumathi, you have presented a beautiful uh, talk on a subject of growing interest in the medical world. I'm sure it holds enormous potential for treatment of certain diseases which still scare the whole humanity. I also get a feeling watching the way you delivered your talk that you are not only passionately involved in the work you are doing, but you have a vision to carry this work forward. And that perhaps might be the reason why you are with a group like us, ours, because this is one way to spread the information about the way you are working on this project, and you have already spent about a decade after obtaining your doctorate. I personally feel that if our friend Chandra Khan Sahab were here, he would have used beautiful expressions to expressions of praise, expressions of gratitude to you.
I
I
welcome.
am I miss and I lack that command of language, but my heart speaks that you are destined to present, to become a legendary researcher
Thank you, sir.
in this field. And having seen you and heard you, I feel God will bless you with spectacular success.
Thank you, sir.
There was a question which you answered very well. How do we compare our stage of learning in this field with advanced countries like US? And you frankly said that we have a long way to go. But this is the way you will be inspiring more people to make an entry into this field of research. I had only heard a success story of stem cell research, now know nothing of how the stem cell treatment is administered, but a close relative of mine in USA, he got cured, or at least he survived. Maybe more than 10 years back, he was treated with stem cell after having been a patient of cancer for quite some time, and he's still not only alive, but he's doing well. So this was something very positive that I had heard about it. And today I feel that it will not be too long before the gap which exists between our standard of knowledge of this field with the advanced countries, with people like you, it will get filled up. The gap will get filled up. I, on behalf of everyone who has listened to your beautiful talk, express my heartfelt gratitude that you took time off to share your learning with us. And I'm sure you will have God's blessings and our good wishes for a
Thank you, sir.
grand success in your mission. Thank you.
Thank you. You made my day. Thank you, sir. Thank you so much.
Thank you.
So thank you. It's a great pleasure of today's
Thank you so
webinar
much, sir.
conclusion. So we will be meeting tomorrow again, and I will request all our participants to take part on tomorrow's webinar, because one expert and one experienced person with very cool and peaceful mind, who will be the chief speaker of tomorrow. We was special guest for today. So tomorrow we will be coming again, and all regard to all senior persons and blessings to juniors also. So we will be meeting tomorrow, and with a hope that tomorrow again we will be learning something. So every webinar should be to learn something and to earn some knowledge from great scholars. Thank you. God bless you. Thank you. We'll be meeting tomorrow.
Thank you,
God
Doctor.
bless
Thank
you.
you, Amitabh ji, and for
Mm.
invitation, Ankit Bhaji, and all others. Thank you very much. Thank you, Doctor.
Thank you. Thank you. Thank you.